Hello, my dear students. How are you all? So, myself, Shaumitra sir. Last time we had already discussed about that of the bacteriology itself. We had discussed about that of different signs lying under that of the bacteria itself. Okay. I'm back again along with IIOCS itself, and in order to like discuss the morphology with that of the molds as well as that of the yeast and that of the actinomycetes itself. Okay. First of all, I would like to ask apology to you all that for keeping you all wait, because actually there were certain kind of technical issues happening, might be from my end itself only, so that because of which I was unable to get connected to you people itself. So now I am okay with the same itself. Okay, and we can start off. So last day, as we had already discussed in the last day itself, like during that of the while we were studying about that of the bacteria, we had already started about that of the studied about that of the like bacterial structure, the bacterial morphology, and all the details things that we had already seen. We had even also seen that what are the different like types of that of the bacterium. Okay, how we are classifying the bacterium on the basis of that of the shapes like cocci, like bacillus, and everything. So in the same direction, in today's class, we are going to focus over that of the morphology of that of the molds, okay, the yeast as well as the actinomycetes. See, first of all, I would like to let you know all students that these microbes, bacteria, the molds, the yeast, the actinomycetes, all these microbes, they are very important with respect to that of the food sciences system. Okay, because we are utilizing them for what? We are utilizing them for like creating, drafting our foods also by via that of fermentation technologies. Along with that, we have to also know about that of their morphology, about that of their complete biology part also, in order to stop their faster growth rate, because we know that if they are growing in a very faster manner, they can cause, I would say, the most important thing, spoilage, which we'll be discussing in the next class itself accordingly. So today we are going to focus about that of the molds, the yeast, as well as that of the actinomycetes and their morphology part itself in that case. Okay, let's start. First of all, we'll be seeing about that of the molds. So whenever we are saying about that of the molds itself, molds are actually what? These are the fungi. If I'm saying about the molds, these are actually the fungi. So when we are classifying the fungus, when we are elaborating that of the classification of the fungus itself, we do have lots of groups of the fungus. We are having Phycomycetes, we are having Zygomycetes, we are having Ascomycetes, we are having Basidiomycetes, we are having Deuteromycetes itself. So in Phycomycetes, if I'm saying about that of the Phycomycetes itself, in terms of that of the Phycomycetes, we are also representing it as that of the algal fungi. The Phycomycetes, we are also representing it as that of the algal fungi itself. And in the same manner, we are also having that of the Zygomycetes, which we are also representing it as that of the conjugation fungi itself. We are representing it as the conjugation fungi, and especially under which the molds used to form. Okay, the rhizobium, I hope so. Like since when we all are in kid stage itself, since that time only, we always used to see a typical kind of that of the like an image in which a bread is there, and over which there are certain kind of branchings like structures which are remaining present, and all those branchings like structure, they are showing what? They are showing the dispersal of the spores itself in that case, isn't it? I hope so. You all had seen about the similar kind of that of the structure itself in that case. Okay, so those groups, we are representing them as that of the conjugation fungi, and under that one group only we are mainly placing them accordingly. Okay, so if I'm saying about the same itself, now if we are focusing, these molds, they are especially the multinucleated kind of the molds itself, and are very specialized to show the formation of the spores also. Okay, they're very specialized to form that of the spores also in that case. They are multinucleated structures itself in that case. Okay, they are even terrestrial living also. They are found in that of the terrestrial condition with certain amount of that of the moisture, with certain amount of that of the moisture itself. If we are discussing about phycomycetes, that means the algal fungi. The algal fungi, it requires considerable amount of that of the moisture itself. But if I'm saying about the molds, then yeast and all other groups, if I'm saying accordingly, we will always say that they will be always requiring what? They will be always requiring like a very limited number or a limited amount of that of the moisture content itself. They will require, certainly they have to, they are growing under the presence of that of the moisture as well as that of the shady kind of that of the condition itself in that case only. Okay, so if you're saying about the molds, especially in this group, if I'm saying about the molds itself, molds are what actually? These are the filamentous group of the fungi. You all know what is filamentous. That means multiple cells are remaining present, and all those multiple cells, they are all present in that of, I would say that in a like a cylindrical kind of fashions itself. They are all connected over each other, one upon one, like that. They are all connected upon each other itself. So that is a like the filamentous group of the fungus itself. Okay, now obviously they will show that of the fungus hypha itself. Now what is this hypha? If I'm saying, if I'm talking about the molds or any kind of fungus, the basic unit, okay, if I am saying about that of the basic unit itself, that basic unit of the fungus, we are always representing it as the hypha. The basic unit of the fungus, we are always representing it as the hypha, and these hypha only, they will be branching among themselves. All these hypha itself, they will be branching among themselves only, and when they will be branching among themselves itself, they will result to that of formation of that of a typical condition which we are representing it as the mycelium. Now hypha and mycelium, these are what actually, these are the major structures for that of what, these are the major structures for that of, I would say, like that of the fungus. Any kind of fungus it is there, that is always the major structure which is remaining present over here. Okay, so if we are discussing about that of the furthermore features itself, now we have to know that these group of that of the fungus itself, in them they are actually showing septate hypha. These group of the fungus, they are actually showing that of the septate group of the hypha itself. Septate, septa stands for cross walls. All the hypha, as because they are the multicellular and multinucleated ones, so whatever the hypha are remaining present, all these hypha, they are been like separated by that of what, they've been separated by that of certain kind of that of the cross walls itself, which we are representing them as that of the septum itself in that. Okay, fine. So this thing is not required to be discussed because fungus means they are eukaryotic in nature. When we are discussing about that of the fungus stands for that they are the eukaryotic in nature itself. Now cell wall, with the main cell wall, it is chitin. So if I'm asking you all, chitin, it is a polymer of what kind of component? Chitin is a polymer of what kind of component itself? If you're discussing in that case itself, chitin, it is actually what? It is actually the polymer of that of, I would say, like, I would say that a component which is present in that of the bacterial cell wall. A component which is present in that of the bacterial cell wall itself, and that component we are representing it as NAG, N-A-G, N-acetyl glucosamine. So chitin is always the polymer of that of NAG or N-acetyl glucosamine in that case itself accordingly, and the cellulose or that of the both itself in that case. So cell wall, it is consisting of that of chitin, cellulose, or that of the like both component itself present over here. Now they are showing two types of the mycelium. One is the vegetative mycelium. Another one we are representing them as aerial mycelium. Aerial mycelium are the specialized one in order to disperse, I would say that the spores itself. They are the specialized one to throw that of their spores itself. Okay. And vegetative, whenever we are saying the thing named as vegetative itself, vegetative word stands for what? Vegetative word means, like, I would say that bodily part itself. Vegetative word means somatic structures. Okay, so vegetative mycelium stands for what? These are the structures which are present in that of the mycelium. These are the structure which are leading to that of their complete body growth, I would say. And aerial mycelium, if we are discussing about that thing itself in that case, aerial mycelium, that is what, this will be the kind of the structure which after their reproduction, I would say that spores formation will be occurring, and those spores, they will be getting, I would say that, like dispersed throughout that of the external surface itself, will be representing them as the aerial mycelium itself. Okay. Now, if I'm saying about the obligate, these are the obligate aerob. Now, there are, we will discuss sometime later. Okay, there are like based on the oxygen requirement, we can say that there can be many different kind of the other like classifications or categories. Okay, the obligate category, the facultative category, and all these things. If I'm saying about the obligate category, obligate word always stands for fixed, strict. इसका मतलब क्या हो जाता है? इसका मतलब ये हो जाता है कि whatever the organism are remaining present, like if I'm telling about the obligate aerob, in terms of this obligate aerob itself, these organisms are strictly aerobic in nature. ये जो organisms हैं, ये organisms यहाँ पर they are strictly aerobic in nature. They cannot thrive within that of an aerobic environment. कुछ कुछ ऐसे organisms भी होते हैं जिनको हम लोग facultative बोलते हैं. We'll see later onwards. Facultative based on the oxygen presence, based on the nutrient presence, हम लोग उनको facultative word से हम लोग उनको जानते हैं. If I'm saying about the facultative anaerobes means for these organisms, they are basically aerobe, but they can thrive within that of what? Within that of the anaerobic environment, like that. Okay, so these molds, they are obligate aerobes, and they used to grow by that of elongation at apical tips of their hyphae. That means the hyphae is there, and what is happening actually? Their tip, okay, their tip, it is growing. Okay, that is showing multiple division, and the tip is growing, and gradually when the tip is growing, after that this tip will be, I would say that, penetrating within that of the surface itself. This tip, I would say that this tip, it will be gradually over whatever the substratum, over which it is growing, it will be getting penetrated, or it will be like I would say that it will be moving inside or over that of the surface itself within which they are growing that of the part itself. Okay, so that is their specialty, that is their major, their hyphal specialty or their mycelial specialty itself in that case. Okay, so if you're saying further the reproduction of the molds itself, if you're saying molds, they generally used to show three types of that of the spores itself, in which the common kind of that of the spores are the sporangiospores. Okay, the sporangiospores, these used to get formed within that of what? These used to get formed within that of a sac-like structure named as that of the sporangium. Do remember, students, always that whatever the spores in whatever the kind of the sacs in which they are remaining present, all these sacs, we are always naming them as that of the gium. You can say it's gia and like that. The sporangiospores are what actually? These are those specialized kind of the spores, okay, which are growing within the sporangium. Same thing if I'm saying about the conidiospores. Conidiospores are what actually? Conidiospores are the specialized kind of the spores which are growing beyond that of the conidiosporangium itself. They are growing beyond that of the conidiosporangium itself. We are representing that as that of the conidiospores itself. And arthrospores, these are certain kind of the spores which are getting produced, I would say that during that of, I would say, like unfavorable conditions and all these things itself, these will be getting produced itself. So we do have the examples as rhizopus. Okay, in that one, that is a typical zygomycetes. Penicillium, which can be considered under the ascomycetes group also. They are also showing that of the mold-like structure in that case itself. Okay, so we'll be moving faster. So a mold, see, which the thing which we had already discussed, okay, a structure over which there are multiple cottony, bushy like that of the structures which are getting present. We are representing it as that of the molds itself in that case, accordingly. Okay. So now next we are discussing about that of the actinomycetes. Wherever we are getting mycetes word stands for mycelium. Mycetes word stands for that of mycelium itself. And actinos word, it stands for that of filamentous. Actinos word, we can say that the filamentous itself. So mycelium and that of the filamentous structure that is remaining present over here. We are like these are actually typical prokaryotic kind of the organisms itself, okay, which are actually unicellular and gram positive in nature, and they do belong to that of actinomycetes group itself, order itself in that case, okay. They are nutritionally versatile. Do remember always, like prokaryotic group, we always used to say that they are metabolically highly active. They are metabolically, they are always highly active itself in that case, okay. So they are highly nutritionally versatile, and they are producing multiple kind of the spores also. These are always producing multiple. Yes, obviously they will be producing, because as soon as they will be getting any kind of that of unfavorable conditions itself, they will result to the production of that of any kind of that of the spores itself, and within those spores itself they will be showing their divisions, and based on that division itself they will be further, they will be growing also in that case. So nutritionally they are very highly versatile, as well as it produces multiple different kind of the spores itself. So these are the soil organisms. They are typically the soil organisms itself, and we had already discussed they are the gram positive one. So if I'm saying the spores, now this is very important. In actinomycetes, how they are, I would say that how they're showing the formation of the spores itself. In actinomycetes, let's say that an old hyphae is remaining present. Now that old hyphae, it will be showing typically, I would say that for a fragmentation process, okay. Now when it is showing typically the fragmentation process itself, after which I would say that like a certain parts which are getting like separated by the other fragmentation, okay. Now those parts, they will be only getting converted within the other spores itself, okay. So they can be either get formed by either by swelling or endogenous spore formation. Students, can you tell me what is endogenous spore formation? Endogenous spore formation stands for what actually? Endogenous stands for what? I would say the spore used to get formed within that of the cellular part itself. Within that of the cellular part itself, the spores used to get formed itself in that case. That thing we are representing it as the endogenous spore formation in that case. Okay. So if I'm saying about that other thing itself, these hyphae that subdivides into that other spores, okay, the hyphae, they will be mainly subdividing into that other spores itself. Now they can be sheathless. Now what is a sheathless? Without covering, sheath. Wherever we're getting this word, the sheath. In bacteria also, they are having one sheath out of that of their like complete bacterial structure, which we are representing it as glycosheath itself. Glyco stands for carbohydrate and sheath means covering, a sac-like covering itself. So whenever we are getting this sheath part itself, in that case, it can be sheathless or have a sheath itself, okay, which partly remains within that of the spores after that of the fragmentation itself. Whenever the fragmentation is there, so after that the fragmentation, they do remains within the spores itself, okay, and they show their major divisions itself and all these things accordingly as such, okay. So there are three major spore types. There are three major spore types which are remaining present. The first one, it is the arthrospores, okay, which is getting formed by the subdivision of the sheath hypha itself. Then we are having aluriospores itself, sheathless hypha, okay. That means just now I said you people that ऐसा type का hypha जिसके ऊपर में कोई भी बाहर का lining present नहीं है, इस तरीके के hypha को we are representing it as of the sheath itself. So I would say that sheathless hypha, the subdivision, it is showing that the subdivision itself. And then the very important thing, the endospore. Last day only we had discussed, I hope so you all are remembering how the endospores are getting formed. Simply a single cell is there, all the protoplasmic content, whatever it is remaining present, all these protoplasmic content during the unfavorable conditions like antibiotics presence and all these things itself, वो क्या हो जाते हैं, they all gets aggregated at the core of the cell itself, okay. They detach themselves from the other cell wall, and when they detach themselves from the other cell wall itself, at that of the core, what it will be doing, at that of the core, it will be, I would say that it will be like dividing, it will be gaining, it will be accumulating the nutrients and all these things itself. That thing we are representing it as of the endospores itself, okay. So that is about that of the actinomycetes. Now we can discuss about yeast, a very, very important component as per to that of food science. Saccharomyces cerevisiae. Actually, there can be lots of group of or genera of that of the yeast itself. Lots of like kind of that of the Saccharomyces can remain present over there. Okay. So we are having what the common group, okay, Baker's yeast, that which we are representing it as, we are representing it as of the Saccharomyces cerevisiae itself in that case. Now, they are forming hyphal syncytia. Wherever we are getting this kind of the word, syn word stands for fusion. Cytia stands for that of cell. Syn word, it stands for the fusion, and cytia, it stands for the cell itself. So these group, these organisms, they are always the true septate hypha. I would say that these organisms, they are always the true septate hypha itself in that case. Okay, that means what they are showing, what they are showing the hyphal connections itself in that case, hyphal presence itself, mycelial presence, and when they are showing the mycelial presence itself, there is the presence of that of the septa. Okay, obviously the septa will remain present, and whatever the septa is remaining present itself, that thing it is represented as true septate hypha itself in that case. Now they are showing two different types of morphotypes, means morphological structure itself. First one is a syncytium, that is the syncytia present, and the second one is a normal one. So in the syncytium, what is getting happened? A single hypha will be consisting of multiple nucleus. Within that of a syncytium, what is getting happened itself, a single hypha will be consisting of that of multiple nucleus itself. That thing we are representing it as a syncytium. And when we are saying about that of the normal, I would say that morphotype, in terms of that of the normal morphotype, a single cell is having single nucleus. Okay, so do remember these things, student. These fungus, they are having two different types of the morphologies, morphotypes, we can say. The first type of the morphotype, we are representing it as the syncytium. Syncytium word stands for that of a single, like I would say that hyphal part is there, and within that single hyphal part, multiple nucleus will be remaining present. That thing we are representing it as syncytium. And the second one, it is always a normal one. When we are saying about normal, it stands for that of what a single cell will be consisting of that of a single nucleus in that case. Okay, so these are dividing by budding or fission. They are mainly dividing by that of their main actually major mode, asexual mode. It is always of the budding itself in that case. Okay. And if I'm asking you people that yeast is belonging to which group of the fungus, I said you Phycomycetes, Zygomycetes, Ascomycetes, Basidiomycetes, and Deuteromycetes. It is always belonging to that of the Ascomycetes or that of the sac fungi, that group itself. Okay. Okay. So if I'm saying yeast, it always shows the formation of the meiospores. It is always showing the formation of the meiospores itself. Okay, and the spore formation, this actually gets occurred by that of the endobudding process itself. The spore formation that will be getting occurred by that of the endobudding process itself in that case. Endobudding. Now, what is that endobudding? You can understand with the name itself, you can understand that a single cell is there, and within that particular single cell, I would say that internally birds and other things will get happen, multiple structures will get formed internally. That thing we are representing it as the endobudding itself. So meiospore formation is getting occurred, spore formation is getting occurred itself, and that is getting occurred by that of the endobudding itself in that case accordingly. Okay, so that thing is remaining present over here. That is a part which we are representing it as the like the basic morphological thing itself. So now we have to know one thing, that this group of the fungus, it is involving both the haploid as well as the other diploid stages. Both the stages, these group of the fungus they are having, and these are getting separated by that of mating as well as meiosis and sporulation events itself. They are getting separated by that of mating, meiosis as well as that of the sporulation kind of the events itself in that case. Okay, so haploid and diploid stages itself, they are remaining present. And like I would say that you can see that I had taken one like image over here, like we can say that this is the budding stage itself. Okay, and here there is a spore formation, multiple endospore or spore formation as getting occurred. This is the sexual reproduction part in which they are showing their mating things also. Actually, Ascomycetes, it shows different methods of the plasmogamy. Ascomycetes, it actually shows different modes of the plasmogamy itself. Okay, where you can say that they can have their primitive, I would say, I'm saying about Ascomycetes, huh? They can, their primitive groups, they can have even flagellated structures also, or flagella presence also. Okay, even like the higher structures, okay, whatever the higher fungus are there, all these higher fungus, they are all, I would say that, missing that afford, they're all missing, I would say, the flagellated structures presence itself in that case. Okay, Basidiomycetes, it is truly now non-flagellated, I would say that one structure. Okay, it is not showing even in gametangial stages also, it is not showing the presence of that of the flagella over there itself in that case. Okay, so we are getting about the hyphae, the pseudo hyphae, and all these parts which are remaining present. So now we'll directly come upon to the microbial growth. That means whatever the microorganisms that we are discussing will directly come upon that of the microbial growth part itself. Now, what is this microbial growth? Microbial growth only stands for that of growth of that of the population itself. Like the population is there, and that growth of that particular population, we are representing it as of the microbial growth itself. There is an increase in the number of the cells. Always there is an increase in the number of the cells itself. That thing we are representing it as the microbial growth itself. Okay, the phases of the growth: lag, log, stationary, and death phase. Okay, the phases of the growth is what we are representing it as lag phase. Now, what are all these phases itself? Actually, wherever we are getting the word lag, lag stands for slow. So let's consider that if I'm taking a single bacterial cell or a single microbial cell, and I am taking that cell and I am placing it within that of what I'm placing it within that of, I would say, like a culture medium. When I'm placing it within that of the culture medium itself, and I'm trying to culture that particular microbe, automatically what will get happen, that microbe will always be taking certain time period to get acclimatized or adapted to that of their environment, है ना? इवन हम लोग भी अगर कहीं जाते हैं तो ऑटोमेटिकली जाने के बाद ही थोड़ी हम लोग ऐसे आराम से हम लोग बैठ जाएंगे, नहीं होता ना वैसा इन दैट केस, isn't it? So the same thing, हम लोग भी कहीं पर जाएंगे, थोड़ा सा बैठेंगे आराम से, बैठने के बाद धीरे धीरे से फैमिली मेंबर्स के साथ एक्लिमेटाइज होंगे, अडेप्टेड होंगे कि भाई यहाँ जब हम लोग आए हुए हैं, we can stay over here, we can talk like in our home itself only in that case. That is the kind of the lag phase itself. बस एक बार जब हम लोग adapted हो गए, फिर तो बक चालू हो जाता है in that case, isn't it? The same thing it gets happened here also for the other microbes. Okay, what is happening for the other microbe also? Whenever I am taking the other microbe and I am like putting the microbes within the other culture media, I am putting the microbes within the other culture media itself. So immediately when I am putting the microbes in the culture media, the microbe will take time to adjust itself to that of their external environment. Now, when it will be taking time to adjust itself to that of the like its external environment itself, that phase we are representing it as another lag phase itself. As soon as it is getting adjusted, automatically automatically that bacteria we will be like, it will be showing what, it will be immediately, it will be getting charged up because it had got adapted to that of its environment. Now as soon as it had got adapted to that of its environment itself, automatically the bacteria will get charged up, it will get excited itself in that case, and automatically afterwards what will get happened itself, automatically afterwards there will be a logarithmic increase in that of their microbial number. There will be a logarithmic increase, or I would say that an exponential increase in that of their, I would say their like the number itself in that case. Okay, that thing we are representing it as the log phase itself. Next, I would say that the stationary phase. Now, what is getting happened for the other stationary phase itself? In terms of the other stationary phase, what is getting happened over here? In terms of the other stationary phase itself, I would say as soon as the microbe is getting into that of the logarithmic phase itself, after which it is showing the utilization of all the nutrients, जैसे जैसे वो न्यूट्रिएंट्स को यूटिलाइज करना चालू करता है, देयर विल बी अ सिंगल टाइम पीरियड, एक ऐसा पीरियड आएगा जिस पीरियड में द ग्रोथ ऑफ दैट ऑफ द माइक्रोब अलोंग विद द अवेलेबिलिटी ऑफ द न्यूट्रिएंट कैन बिकम अ स्टैंड स्टिल पार्ट इटसेल्फ। वो जो पार्ट आ जाएगा दैट विल बी नोन एज अ स्टेशनरी फेज इटसेल्फ। व्हाई इट विल बी कमिंग टू स्टैंड स्टिल? बिकॉज दैट इंडिकेट्स एज सुन एज स्टेशनरी फेज हमारे पास में आता है, वो हमें हमेशा ये इंडिकेट करता है कि as soon as if the microbes are dividing somewhat more or some numbers more, automatically what will be getting happened itself, automatically there will be a declining of that of the cellular number. Because why? Because the available nutrients will get reduced. If the available nutrient is getting reduced within that other part, automatically what will be getting happened? There will be the death of that of the individuals that will be getting occurred over here itself in that case. Okay. So these are the part. We are also representing that as that of the sigmoid shaped curve. We are also representing it as that of the sigmoid shaped curve itself in that case. Okay. So what is getting happened? Number of cells, lag phase, exponential phase, stationary phase, and declining or death phase itself in that case. Time with respect to time, this is happening with respect to the number of the cells, and that is getting, I would say, that expanded over here itself. Okay. Now, how can we measure the number of the microbes? How can we measure the number of that of the microbes itself? So there can be certain indirect measurements like turbidity. What is turbidity? Like we are taking a simple test tube, and within that of the test tube itself, I'm placing that of the like a culture. We are inoculating the culture within the test tube itself. We are like placing that particular test tube for incubation, and as soon as we are placing the particular test tube for that of the, I would say that the like incubation itself, so automatically what will be getting happened? Automatically like when they are showing the division or their increase in that of their number, immediately what will be getting happened? The complete internal solution of that of the test tube will become turbid. Turbid means what? It will be like muddy kind of thing itself. It will be not like a complete transparency will not be getting maintained over here. So by that we can say that the microbe is growing in that case. The one more part, that is metabolic activity. That means what? If there is like, I would say that if in any case the metabolic part or metabolic activity, let's say about the formation of that of an enzyme, it is getting stopped or it is getting raised. Let's say that of the formation of that of the protein or that of the carbohydrates and all these things itself, if it is getting formed or it is getting raised or its concentration is getting raised within that of the culture itself, by seeing that part itself, we can say कि जो माइक्रोब अपने पास में है वो माइक्रोब प्रॉपर ग्रोथ शो कर रहा है या नहीं कर रहा है, that we can say by simply checking on that of the metabolic activities itself. These are the indirect measurements. Now, what are the direct measurements? Plate counts. That is simply we are plating the microbes, and when we are plating the microbes itself, automatically we can say that we will say that it will be always showing that of the, we have to count that of the organism, we have to count that of the cells itself. That thing we are representing it as that of the plate count part itself in that case. Okay. Then I would say filtration. Filtration will also be the part, okay, also be one of the process itself by which I would say that like we can directly measure that of the I would say that that of the organism itself in that case, or that of the part itself in that case accordingly. Okay. So I would say that in this case, like direct counts, if I'm saying about that of the direct count itself in that case, direct count means that you can understand it is directly we are counting that of the organism. So directly we are counting that of the cell itself. When we are directly counting that of the cell, automatically we can say that direct measurement, it can be done over here itself in that case. Okay. So growth factors, there can be like when we are cultivating, I would say that when we are cultivating a particular microbe, there are always the growth factors which are remaining present. Physical factors, pH, acidophiles, alkalophiles, neutrophiles, and all these things, we are representing them as the growth factors which are representing over here itself. Okay. So based on the pH, we see, first of all, we have to maintain the pH. If the organism, if the microbe is loving the acidic environment, then we have to maintain the acidic pH. If the organism is loving an alkaline environment, we have to create that of the alkaline pH itself. And if the organism is loving that of the like neutral environment itself, we have to say that neutrophiles itself in that case. Okay. Then temperatures. If I'm saying about temperatures, the psychrophiles, mesophiles, and thermophiles. If I'm saying about psychrophiles means chill, the temperature, chill temperature or cold temperature, when it is remaining present, we are representing it as psychrophiles. If I'm saying about the mesophiles means what? I would say that moderate range of the temperature itself. Okay, those organisms which are loving moderate range of temperatures, we are representing them as the mesophile part itself. And if I'm saying about the thermophiles itself, what is thermophile? In terms of the thermophile, we can say that it is like heat loving kind of the organism. Those organisms which are loving the temperatures itself or heat itself, we are representing them as the thermophiles itself in that case. Okay, so next we can say that moisture. Okay, presence of the moisture. Obviously moisture is what? Actually moisture, it is always, I would say that, the essential requirement, because unless until moisture is adequate, the organisms I'm sorry, the organisms will not be able to show its proper growth. Unless until the moisture is adequate, the organism will not be able to show that of its proper growth itself. And based on that of the moisture itself only, the organism can be showing that of its proper growth and all the other parts. Then we are having the hydrostatic pressure. Hydrostatic pressure, automatically the pressure itself, the water pressure itself in that case, which is remaining present. We are having tonicity. What is tonicity, students? If you can tell me, tonicity is actually what? Tonicity means concentration. Okay, concentration of the solution. So based on the concentration of solution, we are having hypertonic, hypotonic, as well as isotonic kind of the environment. You all know very well. And in that of the hypertonic environment, what used to get happened itself in the really sorry, in the hypertonic environment itself, the exosmosis from the cell, it used to get happen itself. Exosmosis from that of the cell, it used to generally get happen via that of the hypertonic environment itself. If I'm saying about that of the hypotonic environment, what is hypotonic environment? In that of the hypotonic environment, endosmosis of the cell will get happen, and isotonic environment, what will be there? It will be an equilibrium that will be getting maintained. So hypertonic environment is always related to that of plasmolysis. Hypotonic environment is always related to that of deplasmolysis, and isotonic always helps in maintaining a normalization part itself in that case accordingly. Okay, so radiation, next important growth factor. Radiation, I would say X-ray, gamma rays, these are all the radiations which are found to stop, or I would say that decelerate the rate of the growth of the microbes itself. That thing, it has the radiation itself. Now, based on the oxygen requirement, I was telling you people, strict or obligate anaerobes. Okay, the organisms, we can classify them. Obligate aerobes. We can classify the organism as the facultative anaerobes. We can classify the organism as aerotolerant also, and microaerophiles also. We can classify them. So obligate anaerobes and obligate aerobes. If I'm saying obligate anaerobes means what? These organisms, they are strictly anaerobic in their nature. Obligate aerobe stands for what? These are those organisms which are strictly aerobic in the nature itself. If I'm saying about the facultative anaerobes, in terms of the facultative anaerobes, what is getting happened? These organisms, they are actually aerobe, but they can live within I would say that the anaerobic environmental condition also. So facultative anaerobes are remaining present. Aerotolerant, those organisms which are capable enough to live in the air. Okay, and microaerophiles, which are always requiring like minor or trace air component or air like components in that case. Okay, now certain mineral as well as organic and inorganic requirements which are required for that of the microbes to grow. First of all, carbon. Obviously, carbon is required because the body is made up of the hydrocarbon part itself. So certainly carbon will be, obviously it will be required. Nitrogen, it is certainly essential for the formation of ammonia, amino acid, as well as the proteins itself. Okay. Phosphorus. required for nucleic acid formation in that case. Certain kind of the trace elements like copper, iron, zinc, calcium, and others in that case. Okay, these are all required for electron transport processes, for their respiration part, and all the other details itself. Sulfur, it is also the component of that of an amino acid itself, and vitamins, obviously, it will be required. So these are the major nutritional or biochemical factors which will be required in order to grow a microbial, we can say that organisms itself in that case, or in order to show the growth of that of the microbial organisms in that case itself according. So what we are discussing now, actually all these components. Now when we are discussing let's say that you got like a certain bacterial culture. Now when we got that of the bacterial culture, we are now trying to cultivate those bacteria within that of what, within that of our lab itself. When we are trying to cultivate the bacteria either via streaking method or something like that, first of all we have to create the media. And when we are creating that media itself, okay, it is common for any kind of the cell. It can be that of the plant cells itself, it can be the microbial cells or anything. It is common for any kind of the cells itself. Whatever the kind of media we are taking, we have to incorporate lots of important factors within that of those media itself in that case. Okay, and what are those important factors? They can be moisture, they can be like we have to maintain about the pH, the acidic condition or alkaline condition. We have to maintain about carbon, nitrogen, all these inorganic as well as organic resources that we have to maintain over here. Okay, fine. So now the culture media, if I'm saying the culture media, it will be of, I would say that synthetic media and complex media. Synthetic media, that means what, the kind of the media which we are creating. And complex media, it stands for what, it is the kind of the media which is like made up of that of multiple other resources itself, or multiple other kind of the media itself in that case. That thing we are representing it as. So synthetic media and complex media, the agar, the nutrient broth, and all these things itself. How we can obtain a pure culture of that particular organism itself? It can be always obtained by a special method, and that special method we are representing it as that of the strict plate method itself. We are representing it as that of the strict plate method itself in that case. Okay, and those strict plate method, I would say that like we are taking that of the inoculum and we are like simply streaking over that of the like media itself in that case accordingly, and we are showing the growth of that of the organism. Okay, so whenever we are considering about any kind of microbes, our first and foremost concern is always to like grow them in obviously in vitro condition. Unless until we are growing these microbes, we will be unable to understand the like growth pattern of these microbes itself. And if you're not able to understand the growth pattern of the microbes itself, automatically I would say that we'll be not able to understand that how they are causing the spoilage, how they are causing the foodborne illness, and all the parts itself in that case. Okay, students. So today we'll be discussing till here only. Okay. Next day will be a very important day, because we are going to discuss about many of the lots of important sections. Okay, what are those important sections? We are going to discuss about gram staining. We're going to discuss about that of what we're going to discuss about different types of the bacterium and their differences on the basis of the gram stain. And even we will also touch the food spoilage and the responsible factors behind the same itself in that case. Okay, students. So I would say that we are always with you. IIOCS is always with you, and any kind of support or anything, contact your, I would say that class monitor itself in that case. For the study materials, for anything, you can simply WhatsApp to the given number present over here itself. Only the thing is that prepare yourself to the best and practice a lot. Practice a lot with your concepts, whatever it is remaining present. Obviously, you will be getting a desired, I would say that, your colleges, or I would say that like desired rankings within that of the ASRB net itself. Thank you, everyone. Thank you very much. Meeting you soon, very soon, I would say. Thank you, everybody.